The name of this dissertation defense, the dissertation is called Nutrigenetics of Polyunsaturated Fatty Acids and Vegetarianism. So let's get started. So what is nutrigenetics? So identifying a phenotypic, an observable trait response to a dietary intake that is determined by the genotype, genetic makeup of an individual. And we could think of a therapeutic goal, this overall, what are we building toward here? And it would be to create targeted dietary recommendations for disease treatment and prevention based on an individual's unique genetics. When you hear nutrigenetics, there's also these related terms, nutrigenomics, which, as the name implies, is similar, except instead of looking at the genetic level, we're looking at the genomic level and the epigenomic level, as well as personalized nutrition, precision nutrition, and tailored nutrition. And I'll go through those terms in a moment. There is a growing interest in nutrigenetics. So over the past 20 years, we could actually say that this is a fairly new field. And over the past 20 years, we've seen what's essentially a linear increase in the number of publications, which is great. There's a growing interest. It's a burgeoning new field. And when we're looking at the applications of nutrigenetics, it's useful to think of them in terms of resolution. So we could start at the lowest resolution. So this would be like recommendations for large effect conditions. So I took some terms from Ordovas 2018, but these are all very new terms. So there's no real consensus on what these terms exactly mean at this point. But I like this definition, so I used it. So tailored nutrition, these broad scale things like PKU, lactose intolerance. We have a straightforward implementation, right? Don't eat dairy, don't eat phenylalanine, right? And it's commonly used and employed effectively today. So this is already happening. And then we can think of medium resolution, so recommendations for these small to medium effect nutritional susceptibilities. So we could call this personalized nutrition, things like, take a general demographic group. So for example, European women age 45, right? And they have a mutation at minor allele RS1234, right? And maybe they're at increased risk of iron deficiency because this is a mutation in one of the iron enzymes, right? So this would be like a medium resolution. And then the high resolution, this is like the dream, is a quantitative or even algorithmic understanding of the complex relationships in the human organism. We could call this precision nutrition. So an individual, Mary Beth Johnson with haplotypes A, B, C, D should eat 42% carbs, 25% fat, supplement this, don't supplement that. Maybe if you're exercising, you should have exactly this much more, right? So that's kind of like the pipe dream. But as of right now, in my estimation, I think we're right about here, right? So we're just entering this medium resolution domain of nutrigenetics. And there are some challenges in implementation. And especially there's some challenges to get to that high resolution understanding. For one thing, we're dealing with small effect sizes and variance, right? So I'll talk about this in the context of GWAS in a minute, but we're looking at variants that have small effects. So there's not one gene that dictates what you should eat, right? There's many small effects throughout the genome. And these biobank scale data sets have actually only recently emerged with adequate power to get to this medium resolution, right? I'm thinking of UK Biobank. It's like we're just getting enough power to get to that level. There's also been few randomized control trials to date in terms of nutrigenetics research, although I hope that changes in the near future. And then there's these hidden factors which can confound nutrigenetics, making it even more complicated. Microbiome, resting metabolic rate, stress, breathing, right? Environment, maybe you live near some pollution. So there's a million things, right? And when I was studying nutrition in my master's, there's a phrase which is studying nutrition or optimizing nutrition is like trying to balance an 8-sided teeter-totter. It's like you have more of one thing and then you're having less of another, and it's this very delicate balancing act. So in terms of the nutrigenetics development pipeline, I kind of think of this similar to the drug development pipeline. And we could think of, from the beginning to actually being used by people in the population, right? So disease characterization, this would be like ICD-10, right? You have to know what the disease is before you can study it. And then variant discovery, which is where we live. And then we pass our results to maybe some cellular studies or some animal studies. And then eventually, once you get validation in that way, you can go to randomized control trials. And finally, maybe the FDA is going to say, hey, you should do X, Y, Z, right? But we live right here in variant discovery. And if we want to zoom in to variant discovery a little bit more, we could think of the steps, which are collecting the genotype and phenotype data. And this is the one part that we didn't do. This was done by UK Biobank, so it's really cool. So we received that, and then we did the rest of this. So this is what I've been doing, is designing analyses, quality controlling the data, conducting statistical tests, replicating and meta-analyzing the results, and then doing some follow-up, putting our results into context. And so I'll introduce this tool, UK Biobank, now because it's what we used as the primary data set for these studies. So UK Biobank is a publicly available health research database. It's fairly new, only the last 3 years. And it collected the health status and genotype of 500,000 volunteer participants. They were aged between 40 and 69 years old in the years that this was active, 2006 to 2010. 88% white British, which is unfortunate limitation, right? So these results are really mostly only generalizable to Caucasian population. This is a general problem in quantitative genetics, which is being fixed currently. And then one really cool thing for us as nutrigenetics researchers is that UK Biobank has 2 dietary surveys. They have a food frequency questionnaire that they gave at the initial assessment, and a 24-hour recall survey, which was administered in voluntary waves to these participants. So that's our data set. Let's start to talk about GWAS and the theory behind that. So we have what's known as the common disease, common variant hypothesis. And I say this every time, that people in my lab have seen me explain this at least 20 times, but I'll do it one more time today. So the idea is that the genetic component of common disorders is due to a large number of disease-causing alleles that occur often in the population. So in contrast to a Mendelian mutation in the upper left quadrant, things like cystic fibrosis, cleft chin is one of these, where it's like one gene is directly impacting a phenotype. We're more interested in this lower right-hand quadrant, where we have many small effect variants throughout the genome that are sort of giving this cumulative phenotype, right? So things like some certain types of cancer, diabetes, obesity. This is why you never hear of the obesity gene, right? There's no one spot in the DNA that makes you obese or susceptible to that. But what we have is, we can develop a polygenic risk score with, let's say, 80 variants that are making some contributions to obesity throughout the genome. And so the tools we used were GWAS and GWIS. These are related statistical analysis techniques to identify significant nutrigenetic variants. These are single nucleotide polymorphisms when they're a single base pair of DNA. These are called SNPs, but I use the term variant because we're also talking about indels, which can be multiple bases, right? And genome-wide association studies are used to find associations of allele frequencies with disease traits. And we're interested in the effect or the odds ratio or the p-value of a variant, whereas in an genome-wide interaction study, we're really interested in these gene-environment interactions and the effect or the odds ratio or the p-value at the interaction. So what does all that mean? Well, it's really, the statistics are not incredibly complicated. We're really just doing about 1 million regression models for each spot at the DNA. So for every base pair that's been genotyped, we're running a regression model. And so we could think of it as the trait of interest, Y would be like the dependent variable in the model. And then we could code the minor alleles. So remember, you have 2 copies of DNA, right? So you can either have 0, 1, or 2 copies of a minor allele, throw some covariates in your model, and that is your GWAS model in a nutshell. For GWIS, we are adding this sort of higher order statistical interaction term where we can, take what would have been an ordinary covariate and call it the environmental exposure. So, maybe this is the dietary intake of interest or, whatever, and then look at the interaction between that and the genotype term in the regression. So that is how they work. And so like I said, we're interested in this beta GG in GWAS, so the effect of the genotype term, and then beta gene environment G by E, so the effect of the interaction, right? So in terms of gene environment interactions, there's actually many different ways that these could happen biologically. So for example, your genetic variant influences the enzymatic metabolic efficiency of a food or a toxin, or a variant modifies the risk benefit of a behavioral exposure like exercise. You could think this person only exercises for 2 hours a week and they're, ripped, and I exercise, double that, but I, whatever, right? And then you could think of another way, which is an environmental exposure modifies gene expression epigenetically. So this is actually what PUFAs do, right? So PUFAs can epigenetically impact gene expression. So there's a lot of different angles that these interactions can happen. I may be biased, but I think that diet is the most important environmental exposure to study. And I think that because of two reasons. For one, it's a fully modifiable factor, right? So you can completely, within limits, but it's really modifiable. You could change it a lot. And it also, the food you eat directly interfaces on a cellular level with the organism, right? So it's this really direct impact that we have essentially full control over. So I think it's a really nice field to study gene environment interactions. Our tool for visualizing the results of a GWAS and a GWIS is the Manhattan plot. This is the main visualization tool for finding these results. And so on the x-axis, we are looking at every variant by chromosome that has been genotyped. And on the y-axis, we have the negative log of the p-values. So in short, the higher the peak goes, the higher the points go, the more significant these points are going to be. This red line indicates our typical genome-wide significance threshold of 5 × 10⁻⁸. And this is derived from the classic alpha level of 0.05 divided by 1 million LD blocks throughout the genome for multiple testing. We can zoom into one of these peaks and get a regional Manhattan plot. So we can kind of determine exactly which variants are involved in one of these peaks. And so in this case, we have a lot of genetic variants that are in linkage disequilibrium with each other. And so this is kind of the struggle of doing GWAS, is that you're really running many regression equations, but the variables in the regressions are highly correlated with each other, right? So this is what we're doing. So we think it could be the, in terms of causal variant, we think it's the highest p-value, but maybe it's not, right? So we have to kind of consider this entire block, this LD block. And when it's a significant block like this, we can call it a genomic risk locus. Typically presented with Manhattan plots are these QQ plots, quantile plots. And so these allow us to visualize the distribution of the p-values as compared to a chi-squared distribution. So essentially what we can see in a QQ plot is whether we have systematic inflation in our p-values or whether it's following our expected distribution. So these are also typically shown with the Manhattan plot. So the diagonal is our expectation under the null hypothesis. And then above the line would be either true causal effects or inflation, right? And this is something we have to deal with as researchers. So that's it. So I'll get into this first study. This is a genome-wide association study of fish oil supplementation on lipid traits in 81,000 individuals, reveals new gene-diet interaction loci. Catchy, right? So this paper was really fun when it came out because it got a ton of media attention. And actually, if you do the math, so this is 75,000 out of 22 million. So this is actually in the top 0.34% of studies in terms of media attention, which was a pretty, was it fun, Kai? Was it fun during this time period? So I think it was fun, although we got attention for kind of the wrong reason, right? So what the media did is kind of play this game of telephone with our results. And it ended up being, it's a major side effect of taking fish oil, as if we did some kind of clinical study on fish oil and it's bad for people, right? So pretty dramatic misinterpretation of our results, but nonetheless, what do they say in showbiz, right? Any publicity is good publicity, right? So I found it to be enjoyable, to say the least. We released this cautionary note on our results and we said, hey, no, like this is not a randomized control trial. So we tried to clarify that and put out that fire. I will mention here that throughout my PhD, we have had more successful science outreach moments than that moment. So for example, this was one of my mentees, Nuno, and Kaishang and I taught him all about GWAS, and he ended up making this video on YouTube that if you just type in GWAS to YouTube, it's the #1 video on YouTube. It's the first thing that pops up. So that's from our lab, and we're really proud of him. Nuno is a genius that made this amazing video. And then we also have this video about changing DNA into music that has a couple thousand likes and won the second place in the science contest a few years ago. And it's the first thing that comes up if you type change DNA to music, because I know you're at home typing this into your search bar, right? So anyway, just seemed like a good moment to bring that up. So in terms of dietary fatty acids, in this study, we're looking at fish oil, right? So we can separate our fatty acids into saturated and unsaturated. We can further divide into monounsaturated, polyunsaturated. When we're thinking about fish oil, we're really thinking about these two components of omega-3 fatty acids. So EPA and DHA are the major components in fish oil, right? Fish oil has a broad array of effects on the human organism. In this study, we focused primarily on cardiovascular function, but fish oil can also affect cognitive function, microbiome health, mood and depression, all kinds of things. It's a pretty impactful supplement. So when we're thinking about cardiovascular function, we can look at dyslipidemia, which are imbalances in LDL, HDL, triglycerides, total cholesterol, and it's a predictive factor for cardiovascular disease and type 2 diabetes. And what's been shown in previous studies is that using these dietary supplements instead of pharmaceuticals to manage dyslipidemia, a lot of studies have shown that fish oil performs just as well as some of these pharmaceutical compounds. So we're getting the same effects, but no side effects from the fish oil, or at least fewer, right? So this is really worth digging into for people's health. And the EPA and DHA supplied by fish oil are often beneficial for hyperlipemics, so people with too high blood lipids. And previous gene fish oil interactions on lipids were sparse and inconsistent. This was, I call it the pre-biobank era of interactions, right? So you're looking at sample sizes of only a couple thousand, and it's really hard to, not only get adequate power in these pre-biobank studies, but also if you want to meta-analyze them, well, this trial was for 6 weeks, this was for, 2 years. So there's a lot of heterogeneity there. So it was hard to get a read on exactly what the genetic impacts of fish oil or genetic interactions of fish oil were. So we did our genome-wide interaction study. We used 74,000 UK Biobank participants, and we employed a 3-stage discovery, replication, meta-analysis model. It's a typical GWAS and GWIS model. So our replication data set was the ERIC study. So that's another 7,000 participants that we were able to replicate our results with. Again, the UK Biobank is really nice for measuring these exposures because it has 2 dietary surveys. So we use data from both the food frequency questionnaire at the initial assessment, as well as from these 24-hour recall follow-up surveys. And in order to do that, to use those 2 data points, we had to discard a fairly large number of people. So for example, these guys in the yes, no, they said they took fish oil supplements, but then maybe a year later when they were followed up with, they said they didn't take fish oil supplements, right? And conversely with the no, yes. So we wanted to get a really accurate read on, are these people supplementing or aren't they? So we looked for the 2 data points. We looked for yes, yes, and no, no. And this is because more dietary assessments are more reliable, right? It's a pretty intuitive thing, but it's also been shown experimentally. This paper was actually looking at a mean of 4 dietary assessments versus 1, and we're almost doubling our reliability, right? So you can see that with 2 time points, we're, if I had to estimate, well, it's somewhere between, what is this, an 80% increase from 0.3 to 0.5. So somewhere between 0 and 80%, right? So it's not nothing. So I won't go through all these numbers, but our 3-stage, we did the discovery in UK Biobank, and we found a bunch of associations that crossed our suggestive significance threshold. 26,000 associations we sent to our replication stage in ERIC. From here, we found 2,100 of those had P less than 0.5 in ERIC, so we sent those to meta-analysis. And we ended up finding only a handful of loci, that would be this right here, that had a significant, what's known as 1 degree of freedom, or interaction without jointly analyzing both the genetic impacts and the interaction. We're only looking at the genetic effect, and we found 4 loci associated with these phenotypes that were significant at 5 times 10 to the negative 8. Of these 4 loci, only 1 had a significant p-value and not a significant main genetic effect. And that was this one right here. For triglycerides, we're looking at RS112, et cetera, which had a significant interaction p-value of 5.7 times 10 to the negative 10, and a very not significant p-value for a regular GWAS model of 0.6. So this is the one that we drilled down into, and we were very lucky to find this one. And so RS112 has a significant interaction with fish oil on triglycerides. This is the regional Manhattan plot where you can see the LD block of variants that are involved in this locus. And we're seeing that these points are centered around this connexin gene cluster, this GJA, GJB2, GJB6. And I'm just moving this so I can look at my notes about connexin genes. So these are responsible for forming hemichannels across gap junctions to enable messenger molecules across cells. So they're involved in cellular signaling. And what was really cool and really encouraging to us is that connexin activity has previously been shown in mouse experiments to change upon exposure to omega-3s. So this is actually a somewhat observable phenomenon that these connexin proteins are responding to omega-3s, although we are the first to show that this connection in humans. And so this significant interaction signal, essentially, like I said, we have a very significant interaction term and a not so significant main genetic term. The joint effect was not so significant. And in a stratified model of taking, so effect E equals 0 would be no fish oil group, E equals 1 is the fish oil group. We can see that if we stratify those groups, we're hitting this SNP is getting a much more significant p-value in the fish oil group rather than the not fish oil group. And we plotted the levels of triglycerides in these what are essentially 6 groups, right? So we have a AA, which is the homogeneous major allele, and then the heterozygote, and then the homogeneous minor allele, which had too few people to analyze. But what we're seeing is that it has a significant triglyceride lowering effect in those who supplement fish oil versus those who don't, when considering the AA versus AG. Triglycerides were significantly higher in the AG heterozygotes who did not take fish oil versus those who did, right? So in the non-fish oil people, the heterozygote is going up, whereas in the fish oil people, it's going down, right? Everybody sees that? Okay, cool. Similarly, for the AA, we're seeing that the fish oil supplementation is associated with lower triglycerides in the heterozygote, but higher in the homozygote in the fish oil group, right? Okay, so to sum that up, we did the first genome-wide interaction study for fish oil supplementation on blood lipids. We found 4 novel interaction loci, which had never been associated with fish oil interactions before, including one which showed exclusively significant interaction effects. And we found a plausible biological mechanism with these connexin proteins at this locus. And our study also provides further evidence that, so I didn't mention this earlier, but there's a lot of inconsistency in these fish oil intervention studies. So when you're comparing 2 groups, it's like, why did this group benefit from fish oil when this one didn't? Why did this study benefit when this one didn't? Well, we're adding to this pile of evidence that says that genetics are playing a role in this response to fish oil. Okay, so that's that study. Study number 2, 51 novel and replicated GWAS loci for polyunsaturated and monounsaturated fatty acids in 124,000 Europeans. Currently in revision. So again, we'll go to our handy dietary fatty acid chart, but in this case, we're looking at the whole domain of unsaturated fatty acids. We're looking at monounsaturated as well as, so MUFAs as well as PUFAs, and several subclasses of those. So I'll introduce PUFAs as a class. These are dietary fats that contain 2 to 6 double bonds along linear carbon chains from 14 to 22 carbons in length. Imbalances in PUFAs are involved in a broad array of diseases, including cardiovascular disease, cancer, depression, dementia, and a lot of general inflammatory processes. And, you know, if you study biology, you know that inflammation is kind of this wide-reaching phenomenon that affects a lot of disease phenotypes, right? So if you're affecting inflammation, you're affecting a lot of stuff. Not to say that every single disease is inflammatory in nature, but it does matter a lot. And the 2 most common classes of PUFAs are the famous omega-3s and omega-6s. And so omega-3s have all these benefits. They've been shown to improve metabolic syndrome related to obesity, insulin resistance, hypertension, dyslipidemia, whereas the results for omega-6s have been mixed. It's been, sometimes they find positive, sometimes they find negative. These are the so-called seed oils, right? And a proper ratio of omega-6 to omega-3 is recommended for optimal health. Recommended ratio between 1 to 1 and 2 to 1. But what is the current ratio in today's American? See if anyone was paying attention last time I presented.
1 to 6.
Any other guesses? It's even worse. It's even worse. We're looking at 15 to 1 or more. Now, you may say, well, who cares, right? But in terms of nutrient imbalance in the past 100 years, since, let's say, the Industrial Revolution, this is one of the major, I think, nutrient imbalances that has occurred from the processing of food, primarily. So this is an important thing to study. We can also look at MUFAs. These are dietary fats containing, as you may have guessed, 1 double bond along their carbon chains. Largest meta-analysis of MUFAs in 2022 found the highest MUFA intake group versus the lowest had an inverse association with all-cause mortality. So it seems to have a protective effect against mortality. But there have not been reliable associations with cardiovascular disease or cancer or any really subclass of disease risk. So we know a lot less about MUFAs than we do about PUFAs. They're less well studied than especially omega-3s and omega-6s are. And we could think of like common oils and things like that. So, you know, butter is a saturated fat predominant oil, but olive oil is the classic example of MUFA, right? So these got omega-9 oleic acid predominating in olive oil. And then many of these other oils are kind of a mix of MUFAs and PUFAs. You see canola oil, very typically used in restaurants. And so, yeah, so a lot of times when we're dealing with oils, we're looking at a blend of these, right? But of course, if you want omega-3 heavy foods, you can talk about salmon, fish oil. Those are the two. So results from previous GWAS studies indicate a strong heritable component is involved in determining PUFA levels. And this NMR enables, for the first time, we're starting to be able to do this large scale quantification of these biomarkers, including PUFAs and MUFAs, these blood biomarkers. And so the UK Biobank recently released what is probably, I think, the largest NMR data set of these biomarkers ever, I want to say, or at least one of the greatest, for over 100,000 participants. So we took all of those phenotypes that were in UK Biobank, all the unsaturated fatty acid phenotypes. I'm not going to go through each one of these, but I will point out the 5 that we were able to replicate in external data sets besides UK Biobank are these 5. So these are the ones that we're most interested in: omega-3, omega-6, DHA, which is a type of omega-3. It's a class of omega-3. LA, linoleic acid, is a type of omega-6. And that, oh, and actually this should be MUFAs. Sorry, I just did this. This should be here. And total MUFAs. Okay, so this is the total amount of stuff that we did in this study. It's essentially on the left-hand side, a 3-stage, again, GWAS. We've got the discovery in UK Biobank, as well as replication in 2 external European cohorts, and a meta-analysis of all 3 of those. Additionally, we looked at the multi-ancestry groups in UK Biobank. This includes African, Central, South Asian, and East Asian groups in UK Biobank. We did a ton of follow-up analyses, which we're not going to have time to go through each and every one of these, but I'll go through a few of them. And so let's first focus on just what I call the meat of this paper, is the GWAS and the meta-analysis. These are the big results, right? So we can visualize these results in a circular Manhattan plot. So instead of the typical Manhattan plot, which is horizontal, we're kind of layering 5 Manhattan plots on top of each other. This makes it really nice because we can compare where peaks coincide. Oh, you kind of see the mouse on there. We're comparing where peaks coincide across these phenotypes, because they are very related traits. And we put the novel variants that were found in the UK Biobank, and well, enabled by the UK Biobank, but new loci in this analysis, replicated novel loci, and there are 51 of them across these phenotypes. And what we're seeing in the most significant peaks here are a lot of what we would expect. It's the typical fat metabolism group of genes, right? We've got FADS1 and FADS2, which are the predominant omega-3 and omega-6 metabolism genes. And then we've got these LIPC, LIPG. So a lot of these lipid metabolism enzymes are here. So that's kind of our main result. And we can also look at the results of the multi-ancestry GWAS. And we were able to actually find 4 additional loci in these multi-ancestry groups for the traits DHA percentage of fatty acids, total PUFAs, and that's an DHA percentage again, right? And so we found 2 loci in the African population and 2 loci in the East Asian population, which had not only never been found for these populations, but also had never been found for Europeans in general, or any ancestry group, I should say. Europeans have been, again, the predominant group that have been studied, but they've never been identified at all. So we've got 4 additional groups. So that's pretty cool, right? And what we're seeing is some suggestiveness here that there are a different group of genes involved in different ancestries' metabolism of these nutrients, right? Finally, I'll go through this tissue expression analysis and gene set analysis because it helps put our results into a broader context. Okay, so for these 5 meta-analyzed traits, we got this really nice result, which is all of the variants, all of the genes at the significant variants for these traits are significantly enriched in liver, significantly expressed and enriched in liver tissue, and actually exclusively in liver tissue. So this may be hard to read, but the red bar in each of these plots represents liver tissue, and it's the only one that's significant. I don't know if I should be asking the audience. It's my nature to ask the audience questions, but I think I'll just answer my own because I'm defending my dissertation. But why is liver the one that we're getting, right? And it's because this is where the majority of PUFA and MUFA metabolism is taking place, right? This is the site of the metabolism to a broad extent. So that's perfect, right? We can also look at gene set enrichment analyses. So genes map to significant SNPs. Oh, okay. So we mapped, we received genes that were mapped to the significant SNPs in the analysis via position, so the nearby genes, as well as those that were mapped via liver eQTLs and liver chromatin data. So we got a big basket of genes that were mapped to our significant results. And so first we looked at this wiki pathways, which can kind of help you put into context like what other pathways have a significant overlap of genes with these significant PUFA and MUFA genes, map genes. So we're finding a lot of other fat related things. So statin pathways, also an epigenetic pathway is fairly high here, the histone modifications, metabolic pathways of our friends LDL, HDL, and triglycerides, as well as some other lipid traits and some vitamins and minerals. So yeah, this is interesting. And we're able to provide this information, say, okay, here's the overlap between PUFA related, these genes that are influencing PUFA levels, as well as these other traits. And we can look at GWAS catalog and see what are the traits that have significant overlap of these genes that have been done in previous GWAS. So triglycerides, LDL, and interestingly, some mental disorders here. Now, omega-3s have been associated with some mental psychological condition, right? Have they? And Huifeng is doing a project on that. And so we're seeing autism, schizophrenia, bipolar, there's one more. General factor of neuroticism, right? So it's very interesting that we're finding these results as well. We can just look at the novel variants in terms of GWAS catalog similarity and enrichment, and ignore that. And what we can find is that there is a interesting recurring signal coming up in this plot that I wanted to zoom into a little bit. Do you guys see it? It's that we're seeing alcohol a bunch of times, right? We're seeing in drinkers, response to alcohol, alcohol use disorder, alcohol consumption. So I was like, okay, let's plug into that a little bit more. So I stratified the results and I said, okay, variants mapped to alcohol use disorder of the known loci, and then variants mapped in the novel loci. And we're seeing that the novel ones are contributing so much to our being able to connect PUFA and MUFA associated genes with alcohol use disorder. So why does that matter? Well, because these have actually been associated in previous studies before. For example, DHA has been shown to be neuroprotective against binge drinking and is depleted with alcohol exposure. Deficiencies in omega-3s are associated with bipolar, which can trigger alcohol cravings. And alcohol abuse has been characterized by a change in this fatty acid ratios, oleic acid to LA. Of course, we need more research to disentangle these relationships, but what we're doing is contributing another, what I would consider a big piece of evidence to show that there is a fairly robust association between PUFAs and alcohol use disorder, which was cool, right? It's interesting. So in conclusion for this one, UK Biobank has enabled discovery of many new variants associated with levels of circulating PUFAs and MUFAs. We've performed the largest GWAS of PUFA and MUFA levels. We've replicated these results in a European sample, 3 cohorts, 124,000 participants. And we also found some new variants associated with non-European populations. And we found some new variants that strengthen the link between PUFAs and alcohol use disorders. So that's that project. I know this is fast, right? This is a lot of information. This is the last study. Is everyone still good? Yeah, everyone's good. Okay. This is the last study. And this one is called Gene Vegetarianism Interactions Detected in Genome-Wide Analyses Across 30 Serum Biomarkers. My friend, who is a graphic designer, made this really cool, it was actually initially a favicon, is that how you pronounce it, for the website where we put the code, but I just use this as the logo of this paper, which is very fun. I would recommend making little logos for your papers. Makes it a lot more fun to present. So vegetarianism. So what is vegetarianism? It actually is a broad term. People mean a lot of different things, as I'll show. And by the way, this presentation is definitely going to go like 5 minutes over time. Leave if you have to, stay if you want to. Please stay. But the predominant definition of vegetarianism is going to be this lacto-ovo vegetarianism. And that's plant-based food plus dairy and eggs. No meat, fish, or seafood. Recent large meta-analyses in the past 3 or 4 years have found health benefits associated with vegetarianism, improved blood lipids, reductions in BMI, heart disease, type 2 diabetes, certain cancers. Interestingly, no significant differences in all-cause mortality have been shown in the recent large meta-analyses. One of the contributions I've tried to make in this paper is to put in one place what I consider a lot of biases in vegetarianism cohort studies. A lot of them are confounded by what us in the biz, right, in the epidemiology field, call information and selection biases. And I'll tell you what those mean. So in terms of information bias, one source of that is heterogeneous and imprecise questionnaire design in defining vegetarianism, which I'll highlight in this study. And one solution to that is using multiple dietary assessments, defining our variables more carefully versus a single question. So we did use multiple surveys in this study. And again, this goes back to the beginning, right? I showed the reliability of multiple surveys versus one. It's much higher in multiple surveys, right? Everybody remember that from earlier. So we also have selection bias. Vegetarians may be more health conscious in general than omnivores, right? So this is called the healthy user effect. So you may be healthy because you're a vegetarian, or you may be a vegetarian because you care about your health, right? You think it's the healthiest thing you could do. So we're not sure about the direction of causality here. And this can lead to overestimating the effect of vegetarianism. One notorious group of this in the American vegetarian cohort studies are these so-called Seventh-day Adventists. And this is a sect of Christianity which practices vegetarianism, but also increase emphasis on exercise, avoidance of tobacco, drugs, alcohol, We're just doing some clean living in general. And so non-SDA vegetarians have consistently shown when these meta-analyses separate out, here's the SDA, here's the non-SDA, we're seeing much smaller and sometimes not significant associations with disease prevention in the non-SDA vegetarians. And one thing I'll add is that in these vegetarian studies that have been conducted in America over the past, let's say 30 or 40 years, more than half of the participants have been SDA, which is wild, right? So we're generalizing and saying vegetarianism is this amazing thing because it was found to be good in the set. Now, I'm not saying vegetarian is not an amazing thing, but I'm saying this is a very biased sample to use for the majority of the studies, which is the current situation. And so matching participants is one way to alleviate this bias. This is what you do in a randomized control trial, but you can also use matching in a observational study to simulate a randomized control trial where your participants of both groups are the same or as close as they can be. So yeah, we did that. And what about genetics? Well, it turns out there's been no consideration of genetics in large epidemiological studies of vegetarianism. Not one. I looked for months. I looked so hard. There's none. Genetics and ancestry are known to play an important role in metabolic processes, i.e. the whole thing I'm talking about, right? So two ways we considered genetics in this study. One is that we went on this little side quest to see if we could find variants that can explain why a person is vegetarian on a genetic level. But what I consider more important is we did a genome-wide interaction study of vegetarianism across these 30 biomarkers. So we said, are there gene vegetarianism interactions that are affecting levels of these traits in certain genotypic groups? Okay, so the first thing we did is we want to get a high quality cohort of vegetarians. So we found all the people in UK Biobank that took the 24-hour recall survey. So there was 5 instances of the 24-hour recall survey. And because we were interested in the biomarker blood draw, which occurred at instance 0, what we did was only use the follow-up surveys that were closest to that blood draw. In other words, if you said you were a vegetarian here at time point 0, and then not here at time point 1, we didn't care about time point 1, we just care about the closest to the blood draw. So again, if you this is your first time taking the, let's say it's 2009 and it's your first time taking the follow-up assessment, you said you were a vegetarian here, great, that meets our criteria. Because it turns out that even though these surveys, these dietary surveys only occurred within a 3-year time period, it turns out that people who were surveyed multiple times, there's a decreasing trend where the more times you were asked, are you a vegetarian, the less times you said every time that you were a vegetarian. So for the people that were asked twice, are you a vegetarian, only about 60% of them, is that halfway between 50 and 62%, whatever, halfway between 50 and 75% of them said that they were a vegetarian. Similarly, if you took all 5 of the dietary surveys, only less than half of these people said that they were a vegetarian every time, right? And then we can look at on the same instance of these surveys, so even looking at the intra-survey, so this is the inter-survey discrepancies, we can look at the intra-survey discrepancies and see that even on the same survey, 10% or more every time of the participants on that survey said that they were vegetarian, but also said that they ate fish or meat or both on the same survey, right? So this is contained within their definition of vegetarianism, that they're freely admitting that they recently, in the last 24 hours, ate fish. Not just ever ate fish, but in the last 24 hours, because that's what kind of survey it is, right? So to me, this is amazing because it's showing just how much noise we have in this vegetarian signal, right? And how careful we have to be in defining who is a vegetarian if we want a true signal. So we ended up using 4 criteria to define vegetarians. we utilized both of these surveys in the UK Biobank. So they had to have self-identified as a vegetarian the first time they took the survey, or vegan. Vegan is interchangeable in this because there's only about 100 vegans. They didn't say they ate meat or fish on that survey, so these people are out, right? And similarly, on the initial assessment, they had to not say that they ate meat or fish. And then they had to say that they didn't have any major dietary changes over the past 5 years. So these are reliable, long-term vegetarians. What we found is out of over 9,000 people who said at least once that they were vegetarians, only about 3,200 made that cut of the 4 criteria. But what we have here is a really nice, really strong signal to noise ratio, which is important for an environmental exposure analysis, because we're trying to do a clean analysis here. So once we subset by the Europeans, we ended up with about 2,300 vegetarians and a lot more non-vegetarians. This is fairly consistent with the proportion of vegetarians in the population, by the way, which is about between 1% and 2%. And we looked at these 30 traits, we plotted them, and there's some apparent differences between vegetarians and non-vegetarians. But when we're thinking about our covariates, so age, sex, BMI, alcohol, smoking, socioeconomic status, and we also looked at the first 5 genetic PCs, we found that they were highly imbalanced between the 2 groups, and I'll show that in a second. But for example, the average age of the non-vegetarians versus the vegetarians were 56 to 52, so the vegetarians were younger, and the vegetarians were also much more likely to be female, 54% to 66% female. So prior to estimating the effects of vegetarianism, and so we're not doing our genetic analysis yet, we're just doing a traditional epidemiological effects analysis of vegetarianism, but we matched each vegetarian to 4 non-vegetarians along these covariates in order to get a more balanced group. So I really like this. This is produced by the software called Match It, and it is a really nice visualization. It's called Love Plot. What we have here is the standardized mean differences. The differences, so for example, this is half of a standard deviation between the means, right? And so what we're finding is we're getting half, so some of these covariates where there's, half of a standard deviation, like age, BMI. So non-vegetarians were much heavier BMI than vegetarians. They were much more likely to be female. They were more likely to drink alcohol, and they were the vegetarians were more affluent, right? So actually a lot of the stereotypes of vegetarians we found were true in terms of this healthy user effect. And so our matched cohort is super, super close. And so we got a really tight sample of people who, at least, it's not perfect, but at least they're the same age, they're the same sex distribution, they're the same BMI distribution, and all this stuff. So that felt good. And you can, this might be hard to read, but you can see that the unmatched non-vegetarians in the orange versus the matched non-vegetarians, it's really affecting how we're perceiving the differences between these groups. So for example, like what's a good example here? Something like EGFR is a great example where the unmatched are much lower, whereas the matched are almost identical to the vegetarians, right? And of course, the sex hormones, because that proper balance of male to female has been achieved, we're getting what looked like a ridiculous difference. You could look at this without the matching naively and say, oh, non-vegetarians have way higher testosterone than vegetarians, but it's because the sexes were super imbalanced between male and female. So this was cool. And then we used the matched group to the non-matched group. Again, I'm sorry if this is a reading test. Hopefully you can see that. So in all these traits, we've got about 2,000 vegetarians to something like 8,000 non-vegetarians matched. And we're finding what I consider a much more reliable effects estimation of vegetarianism. And a lot of these results coincide with our expectations. In fact, I wasn't really shocked by any of these results, which is good. And I will go through the sections in a moment, but in general, the bright green indicates that the p-value exceeds a multiple testing corrected threshold. The dark green is a 0.5 p-value threshold, and the black is not significant, right? So just at a glance, blood lipids, mostly negative. Triglycerides are higher in vegetarians. Wow. Vitamin D, pretty moderate negative effect. The sex hormones are not being affected by vegetarianism. Liver, we get a mixed bag. But kidney, we're seeing much improved kidney markers. Okay, so let me actually not just freestyle here. Let me go through my slides. So vegetarian diet has several hallmark properties that fit with our results. So what are some of the hallmark properties of a vegetarian diet? You've got restricted cholesterol, right? There's no dietary cholesterol in a vegetarian diet. Increased fiber from all those plants. You've got different amino acid profiles and protein bioavailability. So what we're seeing is a negative effect on all of the lipoprotein markers plus total cholesterol, and a negative effect on vitamin D, which is synthesized from cholesterol, right? Because, yeah, they're not intaking dietary cholesterol, so this makes sense. And high fiber is also known to lower cholesterol. So, for example, soluble fiber is one thing that is known to lower cholesterol. That's why you eat your oatmeal, right? So vegetarianism had a positive effect on triglycerides. This is like a red flag. This is not good, right? High triglycerides are bad. And in previous studies, we've seen sometimes vegetarians have been shown to have these higher, sometimes they're not, sometimes they're lower actually. But we can explain this by the lower vitamin D, or conversely, the higher intake of simple carbohydrates that are sometimes present in a vegetarian diet. We found no significant effects on the sex hormones, not even in the sex stratified analysis. I was actually expecting to find some of these for the same reason about vitamin D, which is that testosterone has that sterol backbone, so it's synthesized from cholesterol. But this is consistent with previous findings where the vegetarians do not typically show lower testosterone or higher SHBG testosterone inhibitor. And vegetarianism is associated with improved kidney biomarkers. We are seeing a negative effect of vegetarianism on creatinine and urea, which are byproducts of protein metabolism, and a negative effect on urate, which is a byproduct of purine metabolism, which again, all these things are found in higher amounts of non-vegetarian diets. Marginally significant negative effect on calcium. Calcium levels are regulated again by vitamin D. So vitamin D is a very ubiquitous hormone that's affecting the levels of a lot of these, and it explains, I've tried to explain a lot of results using vitamin D, and I think it makes sense. Okay, so that was the epidemiological analysis. Got two more sections here. This is a really quick one. We did a GWAS of vegetarianism, and we found no significant variance. In other words, we didn't find a genetic basis to determine whether a person would be a vegetarian or not. One interesting finding from this part was that we found this HLA-DVP1 gene, which has actually been associated with cognitive empathy in a previous GWAS. Some might say cognitive empathy is higher in vegetarians. I know that when I was vegetarian for many years, I was highly motivated by empathy for animals. So maybe that's related. Maybe it's just a chance finding, who knows, but we just put that out into the ether for future people to think about. So we did genome-wide interaction studies on 30 biomarkers, and we found exactly one significant variant. And you can imagine my sigh of relief when we found that one significant result, because hey, we have a paper now. So here it is, the one result, and it's located in this LD block. So calcium is the trait, and it's located in this LD block around this. So the highest significance variant is in the C4 open reading frame, whatever. But these open reading frame genes typically don't have a lot of biological background associated with them, but we're seeing it in a block with variants in this MMAA gene, which is very interesting to us, and I'll talk about that in a minute. We also stratified, we ran just a regression to stratify by genotype what the effect of vegetarianism on calcium, stratified by genotype. We found there's a significantly lower calcium in the CC people, where there's a significantly higher calcium in the CT people. Sorry, I shouldn't say a significantly negative effect of vegetarianism on calcium, right? So that vegetarianism term in the regression model. And then the TT, very common. There's too few of the homozygous minor allele to make any distinctions there. But the point estimate was kind of in this upward trend, right? Okay. MMAA, what is that? It turns out MMAA is involved in vitamin B12 metabolism. And at this point, I'm going, whoa, B12, isn't that important for vegetarians? It turns out B12 is the #1 nutritional deficiency of interest in vegetarians, right? So this is starting to make sense. We're getting these puzzle pieces together. And the closest I could get to mapping calcium into this picture is, okay, we have this metabolic pathway of B12. We're going from the intestine, and then we're going into a cell. This could be a liver cell. This could be many different types of cells. But in order to gain entry to the cell, this cobalamin, it has to pass through a calcium-regulated CD320 channel. And then MMAA is located over here in the mitochondria once it gets into the cell. So, and that's the closest I could place calcium to MMAA. And if we look at CD320, there's a ton. There's even more than this. This is from the protein database. And these are all calcium binding sites. So a ton of these, right? MMAA is most highly expressed in liver. This is where the bulk of B12 metabolism takes place. Cool. And again, this is just showing kind of a zoomed in version. We've got the CD320 calcium dependent entry of B12 into the liver cells. So these are the CD320 channels getting activated. They're calcium binding sites on these. And then B12 goes into the mitochondria and meets our friend MMAA. So that's that. The last analysis in this paper and in this dissertation is the gene-based gene vegetarianism interaction. So we aggregated our p-values to the genic regions to see if we could find some additional significant loci. And indeed, we did find 2 significant loci. We found that in the gene RNF168 in testosterone, we found a significant interaction. And we found in EGFR in ZNF 277. And so again, we can make our regional Manhattan plots and see that the majority of variants here are falling in this RNF168 gene. And I ended up liking that gene to place it in biological context. So we kept that one. For this one, this is in an LD block that sort of spans way across ZNF 277, but also DOC4 is in here, and then this one over here. But I ended up using DOC4 to create a plausible biological explanation for that one. So let's go through these 2. So testosterone, and RNF-168, you can look at the tissues where RNF-168 is expressed. Lo and behold, number one by a landslide, the testes. Some of you may know that is the site of production of testosterone. I'm saying, okay, we're starting to make sense here. Highest expression in testes. And in previous studies, we've also seen that RNF-168 deficiency caused decreased spermatogenesis in a mouse study. So this is actually as far as we got. We weren't able to dig that much deeper for these ones, like we did with the calcium one, but we placed, okay, vegetarianism is causing something in RNF-168 to be causing differential levels of testosterone in these groups. And for EGFR and DOC4, so not only did we get, if you remember, very significant signals in the kidney traits in the epidemiological analysis from the effect of vegetarianism, but we also can look in the literature and find that DOC4 has been previously associated with kidney markers such as diastolic blood pressure, type 2 diabetes, and water consumption. So DOC4 has been placed not exactly with EGFR, but with all these other kidney-related markers. So, so that was that. And so I'll just summarize. I'm way over time. Let's wrap this up. One, SNP had a variant level interaction with vegetarianism on calcium at the genome-wide significance level, associated with expression changes in MMAA, whose gene product is in the B12 metabolism pathway. B12 deficiency is the highest nutritional concern in vegetarians, and dietary intake plays a primary role in B12 availability. And CD320 is a calcium-dependent nearby protein in this pathway. It's the gateway. RNF-168 and DOC4 are likely candidate genes. Based on gene expression and experimental evidence, we were able to put them in that realm of making sense functionally to some extent. It was harder to pin down exactly what was going on with those. More evidence is needed to validate these proposals, as there may be many indirect mechanisms. But again, if you remember way back an hour ago to the first slide, we're just in the discovery phase. That's our job, is to say, okay, there's a million places in the genome you could look. Maybe these three are some nice spots to do your experimental studies. So to conclude this paper, we have, first of all, clearly demonstrated the noise in a single criterion definition of vegetarianism, right? Our use of matching in our epidemiological analysis achieved a balance between vegetarians and non-vegetarians, a better balance that has been achieved in most, if not all, analyses and observational studies, large observational studies. There are some tiny randomized control trials, but in terms of our sample size of about 10,000, I think this is the best balance that's been achieved. We, in the GWAS of vegetarianism, we replicated that there's not a genetic basis to determine whether someone is a vegetarian. And then we found our interaction results. They need to be replicated and validated. Notice that this paper does not have replication. It's a double-edged sword. The benefit in our study of being able to get this really tight definition of vegetarianism also made it so that we didn't have an equally rigorous replication data set. But I think the paper still stands on its legs because it's the first evidence of gene vegetarianism interaction ever. And although no results surpassed the strict Bonferroni 5 times 10 to the negative 8 divided by 30, I'll be upfront with you, we did not pass that. It's very hard to get power to get such significant interaction variance. However, I think the paper stands fine on its merits, and it's useful in the exploratory context that GWIS are, right? We're not doing, as Yahoo News got incorrect about our first study, right? This is not a randomized controlled trial. What we're doing is providing discovery in general. So I was going to show this. I'm not going to show it, but please feel free to go to this website that I designed. We've made the entire code for this project 100% transparent. Every sub-step, you can see the code, and there are interactive Manhattan plots where you can click on a dot and it tells you what the thing is. And so this is, I think, another merit of this paper is we have tried to make it as transparent and open source as literally possible using this R Markdown website. Oh, there it is. So go there from my GitHub if you want to. It's a fun website. Okay, I've got one slide of final remarks. And thank you all for staying and hanging out and listening. I know it's a lot of stuff, right? Okay, we could talk about the current state of nutrigenetic developments. Where are we right now in nutrigenetics? There's some good and some bad. Good. We've got open source GWAS and GWIS software, and the data sharing practices as well are rapidly improving. Even since I started my PhD, the software to do these experiments, as well as the transparency of code, I've noticed there's been a noticeable improvement since I joined this lab. That's great. Good job, everybody. And new data sets like All of Us, Million Veterans Program, incorporate not only larger samples, but we'll finally be able to start to get past this European-centric results, which has been dominating quantitative genetics. And that's happening right now. And we'll be seeing those results in the next decade. So great. Nutrigenetics was also central to the NIH 2020 to 2030, what they call the strategic plan for nutrition research. It was nutrigenetics was the core of that whole thing. So cool. We should be seeing a lot more funding. Kai-Shang's lab has been funded for this exact reason, right? Is people are putting money into figuring this stuff out. But there's some bad too, unfortunately. And probably the worst thing is actually the best thing as well, which is that the pipe dream of being able to really figure this stuff out is such a big promise that it's discouraging how slow the realization is happening, right? So we are, I think right here, I've tried to make a contribution and we've gotten a little, microcosm closer to being able to put this whole picture together, but it's a gigantic task, but we can do it. And this so-called Eat Lancet Commission and the World Economic Forum, these large globalist organizations are unfortunately ignoring personalized nutrition, and they are actively promoting carbon footprint diets, universal reference diets, totally not genetic based. They're saying everybody should eat the same thing because it's good for the environment. An acre of corn is less carbon footprint than an acre of livestock. QED, that's the whole picture, right? This is what I call depersonalized nutrition, right? So it's like I'm just sitting here reading these and well, what am I doing here exactly? Like you're actively trying to, ignore the breakthroughs that are being made in this field. And it's not, I'm not making the giant breakthroughs, but people are. This field is developing rapidly, and a lot of good scientists are doing a lot of good research. And yet you have these giant organizations at the Lancet and at the World Economic Forum that are minimizing this paradigm of gene-based nutritional optimization. So that's my 2 cents on that. That's it. Thanks everybody for listening. Acknowledge my funding. Awesome lab. Couldn't have asked to be with a kinder, more intelligent group of people than in our lab, especially this man right here, Dr. Kai-Shang Ye, has been an amazing mentor, an amazing friend, and an inspiration, one of the smartest people I've ever met. Here's my cat. I also had a really great advisor in my master's program, Dr. Greider, who I think is joining us via Zoom. Yeah, he's here. So thank you to Dr. Greider for also being an amazing mentor, amazing person, extremely smart. Really, I've been so blessed to work under both of these guys in my 5.5 years at UGA. And of course, thanks to friends, here's my dad, etc. That's it. Thank you, everybody. I'll take some.
Open up for questions.
Yeah.
Yeah, any questions? For people that are online, just unmute yourself and you can talk. And start from the people to the loop. Questions? Errol? Can you explain more about the matching?
Yeah, it's a software called Match It, and they actually use algorithms based on this thing called the propensity score. So you may have noticed in the love plot, there was this thing called distance that I didn't explain. Let's go back to that slide. So they try to optimize the propensity score of the exposure and the non-exposure groups. But essentially, I mean, they're using some stats, like the they're trying to optimize the Kolgornorov, whatever KD statistic. Can't remember those Russian guys' names, but they're trying to optimize the statistics through an algorithm. But it's pretty similar to just, here's a person, who are our 4 most similar people based on the covariates, right? So yeah, it's really nice software, really simple, and highly recommend if you're doing a research in this context to look into that. Kaishang, I'll let you moderate the questions. Kaishang.
Yeah. Anyone else wants to ask questions?
I have a question.
Yes.
Yeah, this is Kunling. Thanks, Michael.
Hi.
Yeah, very amazing that presentation and
Thank
really
you.
great talk. I really enjoy this presentation. But I have a question about I think probably your second study is about the fish oil that regulate the TG, or it have an effect on the TG. My question is, have you also considered the quantity that the participant must take? Like, will there be like difference if the participant take like 1200 milligram per day or like 600 milligram per day, but that will also affect this TAG amount? Or it's just only like you only need to take or not take?
Unfortunately, we couldn't get any deeper than that resolution of take or not take. I did try to dig in when I was first starting this project and see, is there some kind of typical standardized dosage of fish oil that we can, kind of feel comfortable. And I think it's something like 1200 milligrams EPA. I can't remember the exact numbers, but there is kind of, if you look at the most, I really looked for what's the most commonly sold fish oil, and then what's the quantities, right? But unfortunately, in a study like this, we have to just take that as a imprecision, unfortunately. And yeah, we weren't able to dig any deeper into that, unfortunately, but I did share your concerns.
Okay, because I think there's maybe a more general question about like this nutrition. Like, if someone have like, say, calcium deficiency, this kind of disease, or not disease, just this kind of like this behavior, do you think that if they take like 1200 milligrams like calcium or any other nutrition supplement, this will be better than, I mean, the higher the dose will be much better than the like lower dose? Or do you think might be an optimized amount for them to intake in order to have this kind of benefit effect?
Well, I love this question because it lets me draw on some of my nutrition background. So there is like an upper dose of toxicity for all these supplements that people need to think about. But I am a proponent of, and I'm not a doctor, this is not medical advice, but your body is your own laboratory, and do some things and see what feels good, and your body will tell you if it feels good. And a lot of this stuff, like, I mean, you could take fish oil for a couple of weeks, and I've actually really noticed effects, like physically, like energy, things like that. But aside from that, more esoteric stuff, what I would say is go by the, there's nothing in this dissertation that can recommend dosages, or again, I try to make this really clear because I'm kind of traumatized by how much people got this wrong when we first started doing this. But but really we're here, right? We're in the variant discovery, and there's a lot of down the pipeline to dig down into the dosages.
Okay. Yeah. Thank you. Thank you
Thank you
so
for your
much.
questions. Nice to see you, Kunlun.
Yeah.
Okay. I thought when you were initially explaining the epilogue, I think the first part of the last one, you had said like there's no difference between the vegetarian and non-vegetarian testosterone.
Mm.
So how did you get to like the RF?
Yeah, exactly right. So this is one of the cool things about studying interactions, is that you can actually find for things that don't come up as significant in the traditional epidemiological analysis, you can start to dig into the differences between genetic subgroups, and you can find differences. So that's amazing. So maybe there is, for, if I were to speculate and take a step into the speculation domain, like maybe for people with certain genotypes, their testosterone is affected significantly when they're vegetarian. And maybe for people with certain genotypes, they can absorb the effects of vegetarianism and not be so affected, right? And actually, I mentioned cholesterol a lot, but the majority of cholesterol is actually endogenously synthesized. So there's a small portion that comes from the diet, but the majority of cholesterol is just made in the body. So it could be that people have more or less efficiency of synthesizing that cholesterol, and the people with good cholesterol synthesis efficiency can bear the brunt of vegetarianism, no dietary cholesterol, and not have their sterile stuff affected. Maybe some other people are less good at that, and they need that extra dietary in order to maintain the healthy levels. Yeah. Does that answer your question? Dr. Greider.
Yeah.
Hey, as enjoyed your presentation. I had a question about the database. So, does it within the database, is there information concerning the racial characteristics of individuals, even just among the British? Is it strictly British, and then any indication that it might be, individuals that may have mixed with Saxons or, Germanics, or, that might give insight as to the variance associated with your results with the fish oil?
That's an awesome question. And because population stratification is such an important concern in these types of experiments, we don't want to accidentally attribute a phenotypic difference with a difference that's actually coming from these differences in the genetic composition of two populations. So to answer your question, we used, I think I have this on a slide actually. No, I don't. Oh, I wish I had that. But anyway, we used this project called Pan UK Biobank, and they did a really nice, even better than UK Biobank did, came up with a really tight definition of British European participants. And then on top of that, in addition to only keeping those participants, we controlled for the first 10 principal components, genetic principal components, and use them as covariates in all our analyses in order to correct for some of those minor differences within the subgroups.
Another, I guess it'd be associated with that, maybe. Would there be any possible indications between individuals that were maybe their origins were more associated with Neanderthals versus Homo sapiens?
That's a great question. We weren't able to look in that. I think somebody will be able to look at that in UK Biobank, because the genome sequencing, I mean, if you get your 23andMe done, you could see 1% Neanderthal or whatever. So there are some indications of being able to quantify that. And I think it's probably definitely making an impact, because it's such a major divergence in human populations that you would expect some of the metabolic processes to be different in those two groups. Yeah, but it was beyond the scope of what we did, but it's an interesting point.
Okay.
Great,
so
thanks.
Thank you.
Thank you, Dr. Grider. Thank you for introducing Michael. When we have our first meeting, we are way over time, but I want to ask one last question because Michael, you're done with us, and in your last few years of working on your project, you may come across some ideas that are exciting, but you didn't have time for it.
Mm.
Can you share one or two of those ideas with us?
Oh, sure. Thanks for that. Set me up for such a great question. I mean, this vegetarianism data set that we've come up with, I'm so glad that some of the other lab members, specifically Alex and Armin, are going to be able to further pursue this rigorous definition of vegetarianism that we've developed, because I think that this opens up the door to a lot more analyses. You could look at binary disease traits. You could look at the NMR values for these vegetarians. So there's that, but really UK Biobank, it lets you do so much. I mean, you're talking about a data set where it's like, you know, a 10,000 by 10,000 grid of ways that you could combine these factors, right? So it really, it's as large as your imagination can be. And like this analysis was really something that I felt very passionate about and very happy to be able to do what I wanted to do with the data, which was to really refine these definitions and to really get to the signal rather than the noise. So I hope that the vegetarian route is pursued. In terms of other, you could look at other dietary supplements in the way that we looked at fish oil. There's a lot more dietary supplement data on UK Biobank. It's a regular wonderland for a nutrigenetics researcher in terms of how much you could really do with it. And I feel really lucky to be able to use. I mean, talk about luck, right? It's like the year that I was able to do this, like this just came out, you know. So I'm like so blessed. I feel to be able to have used this resource.
Okay, cool. Thank you. Just a reminder that we're going to have a celebration. We're going to celebrate at 5 p.m. today. Join us.
Yeah.
And let's thank Michael for the great presentation.
Thank you. Thank you. Yeah, everybody come to 5 bar if you want to.